Hello, my name is Rola Hammoud. I'm a postdoctoral research fellow in Dr. Daniel Drucker's lab at the Lunenfeld-Tenenbaum Research Institute at the University of Toronto. I'm excited to give you a quick overview of our research paper published in JCI Insights, showing how gain and loss of GIPR signaling alleviates gut inflammation in mice. This research paper is the product of the hard work of various members of the Drucker Lab, as well as our collaborators at the Tel Aviv University and Sourasky Medical Center and the University of Cambridge. So let's start by introducing our hormones. GIP, or glucose-dependent insulinotropic polypeptide, and GLP-1, or glucagon-like peptide 1, are incretin hormones, meaning they are hormones released from the gut in response to food intake, and act on the respective receptors, GIPR and GLP-1R, on the pancreatic beta cells to potentiate insulin secretion and reduce blood glucose. Beyond the pancreas, GIP and GLP-1 also converge in their action on the brain, reducing appetite and promoting weight loss. Due to its higher efficacy in promoting weight loss and glucose regulation, GLP-1 receptor agonism took center stage as a pharmacotherapy for obesity and type 2 diabetes. GLP-1 agonism also showed beneficial effects beyond weight loss, including reducing liver steatosis, renal protection, and cardioprotective effects. A common mechanism underlying these beneficial effects had to do with the role of GLP-1 in reducing systemic and tissue-specific inflammation. On the other hand, less is known about the extrapancreatic actions of GIP. With the development of new, highly effective weight loss drugs that incorporate the GIP receptor agonism or antagonism alongside GLP-1, there is greater interest in understanding the biological functions of GIP. We know that GIP promotes adipose tissue accretion, regulates glucagon secretion, and bone resorption, but its role in the regulation of inflammation is less understood. So what do we know about GIP and inflammation? We know that GIP receptor is expressed on immune cells derived from the bone marrow, specifically cells of the myeloid lineage such as macrophages and monocytes, and signaling these cells regulates hematopoiesis and myelopoiesis. The GIP receptor signaling also on the macrophage inhibits adipose tissue inflammation in the context of diet-induced obesity. But the role of inflammation in the gut remains unknown. So why the gut? Well, it's the region for the first line of defense against pathogens, and gut health is slightly linked to metabolic health. So we hypothesized that GIP attenuates gut inflammation, and these actions are potentially mediated by bone marrow-derived GIPR-positive myeloid cells. To test this hypothesis, we conducted a series of studies. First, we tested the effect of GIP receptor activation using a GIP receptor agonist alone or a GIP-GLP-1 receptor coagonist or tirzepatide, and wanted to test the effect of these drugs on gut inflammation. Gut inflammation was induced using a cytotoxin known as 5-fluorouracil or 5-FU. We showed that the GIPR activation alleviates 5-FU-induced inflammation in the gut. We then tested the effect of GIP loss of function. We did this by generating mice with a whole body knockout of the GIP receptor. These mice were more sensitized to 5-FU-induced inflammation. We then wanted to explore the role of bone marrow-derived GIPR-positive cells in the regulation of gut inflammation. We did this by irradiating wild-type mice to ablate their bone marrow, and then transplanted them with bone marrow derived from whole-body GIPR knockout mice. This results in mice with a bone marrow-specific knockout of the GIPR. When exposed to 5-FU, these mice did not differ in their response to gut inflammation. We then did the opposite study, by which whole-body knockout mice were irradiated, then transplanted with bone marrow derived from wild-type mice. These mice were protected from 5-FU-induced gut inflammation, meaning the bone marrow-derived GIPR-positive cells partially regulate gut inflammation. However, this is only in the context of a whole-body deletion of the GIP receptor. We later wanted to identify where is the GIP receptor expressed in the gut. Where is it located and to which cell types? So we tested this through a series of cell sorting experiments and with the use of reporter mice. And we were able to narrow it down to the lamina propria of the small bowel. We did some cell sorting and we found that the GIP receptor is enriched in CD146 positive cells. These are cells that are pericytes and endothelial cells, and that the GIP receptor was not expressed in immune cells. However, the putative role of this receptor in the regulation of gut inflammation remains to be explored. To conclude, GIP reduces gut inflammation, and these actions are partially facilitated by GIPR-expressing immune cells. In the gut, the GIP receptor is found on non-immune cells, namely pericytes and endothelial cells, but the role in the control of inflammation remains to be explored. Thank you.